Welcome back. Now, we are going to discuss the molecular mechanism of endospore formation. Stay tuned. The first step is initiation. In initiation, let me tell you one thing, that bacteria is a wise creature. It can detect the changes inside of the cell environment while also outside environment as well. So how they do it? They have specialized enzymes called sensor kinases, and here four types of sensor kinases are involved in the endospore formation, called kin A, B, C and D. Kin A detects the changes in intracellular ATP pool, which is the indicator of energy, while the B detects the starvation signal from outside environment, and it particularly detects the carbon depletion signal. When the carbon source is depleted, this signal will be detected by the kinase B, sensor kinase B. So how it works? When kin B detects the signal, it causes autophosphorylation, and after autophosphorylation, the kin B is now activated. It will transfer the phosphate group to the SPOF. Okay, this is the downstream protein in this process, and this will transfer the phosphate group to SPO0A. Now, if the SPOA concentration is high in the cell, it means that this cell will form endospore, because the endospore decision is based on the concentration of SPO protein, and these are the master regulators, which mean that these proteins control the expression of genes which are involved in the endospore formation. Now come to the stage 2, is asymmetric cell division, which means that the bacteria will divide asymmetrically, the large compartment and the small compartment. This will form here the pre-spore, which is also called the future spore, and the large compartment is the mother cell. So here in this stage, two proteins are important, which are also the master regulators of this process. One is called SynR and one is SynI. SynR, from this name you can also recall it in an exam, there's SynR. It is responsible for the repression, so it will repress the process, it inhibits or repress sporulation, while the SynI is the initiator or the inhibitor of R, the SynR. So this I, it promotes spore formation. So this was a stage 2. Stage 3, which is engulf. Engulfment means that the mother cell in the pre-spore are here. So the mother cell will engulf the pre-spore, and now the pre-spore is inside the mother cell. One is the pre-spore wall here, and the other one is the mother cell wall. This structure, the spore is now become double membrane structure, and the SPO genes are involved in this process because it helps stabilize the structure. In the stage 4, now the spore is inside the mother cell, but it needs protection, so it forms the cortex here. So stage 4 is cortex formation. The cortex formation, the spore produce peptidoglycans with less cross-linking and also remove the water to enhance its resistance against stress. The SPO, VD and other genes are involved at this stage. Now in the stage 5, the cortex is formed, the spore is inside, it forms another layer which is called spore coat. At the 5th stage, the spore coat is formed. There are tough proteins in the spore coat which provide resistance against chemicals and UV radiation, enzymes and other things. In most of the bacterial endospores, spore coat is the outermost layer, while in some cases the bacteria also produce the outermost layer, which is called the exo. This is rare. At the stage 6, you can see here that now the spore is matured, the spore coat is formed, the spore cortex is formed. It undergoes further dehydration and accumulates calcium dipicolonic acid. The most important step here at this stage is that the spore becomes metabolically inactive. Now the spore is completely matured and ready to be released in this stage. So in the last stage, which is the stage 7, the endospore is ready to release, so the mother cell undergoes programmed cell death and release the spore. So we covered 7 stages, but the detailed stage is the initiation stage. If you have got any question, let me know in the comments section. Thanks for watching. See you next time.